Hello, Bismillahirrahmanirrahim. So the first thing about the RNA-seq data analysis, as I told you in the first brief lecture, that there should be a data set, or that data set you should use for your analysis. And then there are two types of data sets. One would be the real-time data you have, like for example, if you have done some experiment and you have sent your RNA or cDNA for RNA-seq, and then you have now real-time FASTQ files, and you want to do the analysis. And the other type would be the secondary data, or the publicly available data. And I've told you previously in my lectures somewhere a lot of times that the amount of data, the huge amount of data that is available on these portals is so immense that if first I would highly recommend any biologist or the researcher to first find the answer of your biological question using the secondary or publicly available data. And when you get, for example, if you want to do the research on Alzheimer disease, or for instance, you want to do research in some infectious disease or in some non-communicable diseases like cancer, and you want to see that, for instance, you want to see the impact of smoking on the gene expression in lung cancer, for example, or you want to see the difference of gene expression in virus affected livers. So for that, now you have a lot of sequencing data, real time data. But if you go to these portals, which I will tell you now, the databases from where you can download a lot of publicly available data for these Alzheimer or lung or liver cancer or disease or HCV or infectious diseases, I'm telling you. And it's almost the impossible that you don't get any resource or publicly available resource to answer the question you have in your mind. And when you first answer that question, or first you define your question and then do the analysis using the publicly available data, and you get some answers, then you can define a new hypothesis on your real time data, because obviously the real time is always the most interesting and important, because you get new information. But first you should go to these resources. So where do you go to find these resources? There are multiple places. Number one, the most widely used is the NCBI Geo, right? And NCBI Geo, I have made tutorials for gene expression analysis, because if you open this website, you will see that this Geo is a public functional genomics data repository, and it has array and sequence based data. Both are available here, and they accept it here. So you can I have made tutorials how to do the geo how to download the geo data set of microarray or expression data, and then do the expression analysis. Also you can do using geo2r. But there is also sequencing based data here. For example, as I told you, like if you search HCC, the liver cancer, and you search, then it will say that there's a lot of data sets related to it. And if you see on the left side, you can see that there are data sets, series, samples, platforms. This thing I have talked in my tutorials. Basically, a series is a study which have a lot of. So one series will have multiple samples. So one study is one series, and then there are different platforms. Obviously, there would be some Agilent, Affymetrix, or for sequencing there will be Illumina and others. And then there's a study type. Here only expression and methylation profiling array is selected. But if you select the customized, you can see there is, if you exclude it, and expression profiling by high throughput sequencing, right? You just click it and you go to, like, to show this data. Then it will reload probably. It should. Okay, like if I reload myself, throughput sequencing data. So maybe they also have high throughput. If I select only mouse, and mouse and only expression, or maybe they have. Yeah, yeah, yeah. Now you can see. I have selected it. Now I have selected it, so it will take me to this. I'm doing it for the first time in front of you, so we both are learning. But I know these resources. So now, for example, there are 603 data sets which are related to expression profiling of high throughput sequencing. High throughput is, RNA-seq. And then if you click any of this study, for example, this study has 2,351 samples. This one has 921 samples. So just to make it a bit, so there are around 31 pages, right? If you go to the next or to the, so for example, single cell analysis reveals cells heterogeneities in hepatocellular carcinoma. The genomic landscape of Mongolian hepatitis. For example, Mongolian hepatitis. You click genetic genomic landscape of Mongolian hepatocellular carcinoma. Now you can see that now there is two options. One is analyze with GO2R, and one is download RNA-seq count. So basically, the RNA-seq count would be the processed counts data you can download from here, and you just click it, and it will start downloading, or it will take you to the yeah. So this is the raw count data. It's just 6 MB, and you if you download it, and then you can use it. I'm not downloading it now. I'm just telling you, maybe in the next tutorials we will go through this. And so here the limitation is that it only has the processed data. But if you look at this one, that SRA, that's I told you about in the previous lecture as well. I was about to take you to this SRA website next. This is also part of NCBI, and you can just go there, and if I open it here, you can see. Yeah. So now this is that will take me to this data set. Here you saw that the total number of samples were around 140 something, and here there are around 142 files. And if you click, for example, this one, so this is one sample ID GSM of it, and here this is the run. And if you click this one, it will take you here, and you can, so basically there is one uh, you can say there is one problem or a limitation with this. The way to download here is that the size are very big here. So what usually happens is that you can either use SRA toolkit. What is SRA toolkit? Basically, SRA toolkit is a small command line uh, tool that is made available by this NCBI, and you just have to use those commands, and in those commands you can uh, download the data simply by just putting the ID. I have made a tutorial earlier. If I take you there and look into that myself for you, SRA. I'll write my name so that maybe it will take me to there. Yeah. So here's one whole tutorial that is of RNA-seq data analysis, and in this you can go here and you can say downloading the sequencing data, and this would take you to. You can follow these commands and you can download the SRA files, which would be you have to convert them then to FASTQ files. Like first you have to download the SRA files, and then you have to download the FASTQ files, convert the SRA files into FASTQ files, and yeah, so that's easy. The steps are mentioned here. So one repository is GeoNCBI, and that GeoNCBI is connected to Geo, and then that is then further connected to SRA that mainly has only the sequencing data. Geo also have the array data, right? So one is a repository to download the RNA-seq data is NCBI Geo and NCBI SRA. Then there is one more. I will just uh, copy the link I have here, and if you see, the other one is the AMBL EBI project. So NCBI is USA based project, and then there's a European project that is AMBL EBI. If you write this ebi.ac.uk, yeah, and that will take you to this European Nucleotide Archive. So here, as I remember, here it's you can directly download the sequencing file, but for example, again I type here HEC, and then encoding, there are around 779, for example, studies. I click it here, so it is taking us to these different oxygen IDs, and then these are different experiments, these are different studies, and if I click study, now it will take me to multiple studies. ERP is the oxygen ID of that. Here you see that genetic mutations of EF2K in HCC, for example, HBV sequence motif associated with HCC in South African adults. So if I click any of the RNAs seek from a collection of HCC models. So now if I click this one, uh, this is a study by Cancer Research UK, and here no public data is associated with this study. So we go back. So keep that in mind. There is always there are always two types of data sets. One would be authorized access, or one would be with no access, and some would be publicly available data. Like for example, here I clicked which one. I go back and check again for you. I was just talking and I click something RNA-seq of HCC, for example. Here, this is one study. I click that. Now it has the study ID, excision ID, experiment run, and then these are the FASTQ files. So these are the raw FASTQ files. You can even download from here as well. Like if you click one, the sample size would be very big. So I will just show, like if you click this one and download selected files, it will then take you to the download option, and it will take a lot of time. So it's not very ideal to download one by one. I would highly recommend you to use the SRA toolkit. But even if you don't have the 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 skills, or you don't have the uh, confidence to do SRA toolkit, then you can just click one by one and download it. So I would so yeah, so that would be a good way out for you. So the last one is, I will go to the last for now. There are other sources as well, but these are the most commonly used. I'm doing it casually here because I just wanted to show you that it's not a big deal to navigate through different browsers. You can just be patient, look for your data. You can easily find the link from where you can download it. So last one, which is the most commonly used in cancer research, that is GDC portal, GDC TCGA dataset. This is a homepage of TCGA. That is, TCGA stands for the Cancer Genome Atlas, that you may already know if you are interested in cancer genomic analysis. So here you can download the cancer RNA-seq data of different types of cancer types. Like these are different types, all these type of the primary sites of different cancer is available here, and each bar represents the frequency. Like even for, like for example, lung cancer, there are 5466 cases already available. So you can explore your data set. The problem here is, the challenge here is that you have to get the access. Maybe some of them have the public access, but most of them you need to fill in the form of the GDC data portal. Then you have to submit it to the link, the email given with that, and then they will give you the access to the data, but it will take a bit of time. But otherwise, you can download the clinical data and other process file from here easily. And yeah, so you have to navigate through this, like And like if I show you, wait, for example, if I write here HCC again, or liver cancer maybe. So there, let's see what comes up. So this is the study ID with this liver cancer. They have array data of methylation of 528 cases, miRNA-seq data of 513 cases, RNA-seq data of 608 cases, and so on and so forth. So you can look for the further details that uh, how you can get the access of them. The one I told you already, that you have to uh, send an application that that I want to download this data, and then they will give you authorized access, and then you can easily download that. So, but things which are available here you can easily download. For example, here now, if you click the repository, and then now here you can see that on the left side there is mRNA-seq data, sequencing, ATAC sequencing data, array data based on different coverages, and here in the tabular form you can see that some of the data is open access and some of the data is controlled access. So I think I'm telling too much of the information, but I would suggest in the end that you should, all of the students who are looking into this video, they should try to download the data using NCBI, GEO, ENA, European Nucleotide Archive, or from the GDC data portal. And when you are get successful, please tell us in the comments that you have downloaded the data. And if you were not able to download the data, please write it down, that what is the challenge you're facing and how I can help you with that. So the best thing I would suggest is to download the data from where? From NCBI or ENA through SRA toolkit, and that would be the most easiest way. These program, those commands are not very much complicated, but you can do it anyways. So, if you have any questions, please ask in the comments. Otherwise, I think that's enough for today. Information you're looking for. See you. Bye bye.